Assalamu alaikum. I am Habibanas from BSPV. Today, my topic of presentation is microbial degradation of plastics. In this topic, we shall see biodegradation of plastic, mechanism, factors affecting biodegradation, mechanisms involved in degradation, plastic degradation pathways in bacteria, biofilm forming microbes involved in degradation, plastic biofilm interaction, bioremediation, and conclude. First of all, we see what is plastic. The word plastic refers to a series of polymer, usually synthetic polymer, made up of chain of carbon-based monomers. Plastic are a wide range of synthetic or semi-synthetic materials that use polymers as a main ingredient. The global market for plastic is growing continuously since their mass production started around the 1950s, but the petroleum based plastics has gradually occupied almost every area of human life. A recent statistical study estimated that humankind has so far produced tremendous amount of plastic, over 8.3 billion tons. Lack of degradability and the closing of landfill sites, as well as growing water and land pollution problems, has led to the concern about plastics. With the excessive use of plastics and increasing pressure being placed on capacities available for the plastic waste disposal, the need for biodegradable plastics and the biodegradation of plastic waste has assumed increasing importance in the last few years. If we talk about plastic, there are two types of plastic. First one is biodegradable plastics, which are composed by the action of living organisms, usually microbes, into water, carbon dioxide, and the biomass. And the second one is petroleum-based plastic, which can be produced by crude oil, natural gas, and petroleum, which can be said as synthetic plastic. Over the last few years, the need for biodegradable plastics has led to the extended significance due to extreme use of plastic and increasing pressure being positioned onto the head capacities for plastic waste disposal. Lack of degradability and the closing of landfill sites, as well as growing water and land pollution, problems has caused a situation about the plastics. Plastics are causing great difficulty in environmental problem, and subsequently, this desires manufacturers to synthesize materials that don't have an impact on the environment. The use of microorganism in the surrounding to multiply the molecular shape of plastic material to produce an inert humus-like material, and this is the much less dangerous to the surrounding. Furthermore, expertise the interaction and the biochemical adjustment so that they undergo are tremendously essential. The next one is biodegradation of plastic. Plastic biodegradation is actually the process of changing properties such as tensile strength and shape, color, chemical structure, molecular weight, etc. This process involves the enzymatic and non-enzymatic hydrolysis of microorganisms, especially bacteria and fungi in the microorganism. Biodegradation is necessary for water-soluble or water-immiscible polymers because they eventually enter streams which can neither be recycled uh, or incinerated. It is important to consider the microbial degradation of plastic and synthetic polymer in order to understand the which is necessary for biodegradation and the mechanism involved. This basically requires the you can say understanding of interaction between material and the microorganism and the biochemical changes involved. Separate studies on the biodegradation of plastic has been carried out in order to overcome the environmental problems associated with the synthetic plastic waste. Ways for biodegradation. There are usually three ways we use in the process of biodegradation. Plastics are generally biodegradable aerobically in nature and aerobically in sediments and landfills and partially aerobically in compost and soil. If we talk about aerobic biodegradation, which is also known as aerobic respiration and is most important part of natural reduction of contaminants in many hazardous waste site. Aerobic microbes use oxygen as an electron acceptor and break down organic chemicals into smaller organic compounds with carbon dioxide and water as a byproduct. In partially aerobic biodegradation, this uses less oxygen and it produces more bioproduct along with water and carbon dioxide. Anaerobic biodegradation is the decomposition of organic contaminants by using microorganisms with the absence of oxygen and is likewise a crucial component of the nature reduction of the contaminants at dangerous waste sites. Some aerobic bacteria use nitrate, sulfate, manganese, iron, CO2 as a their electron receptor to entrap down organic chemicals into smaller compounds. Likewise, if we see the anaerobically breakdown of plastic, it decomposes to methane, carbon dioxide, water, and carbon residual plus biomass. These are the ways for an biodegradation of plastic. And the next one we see mechanism of biodegradation. Microorganisms are not able to move the polymer without delay through their outer cellular membranes into the cells, in which most of the biochemical processes take place. Since polymer molecule and are long and no longer water soluble. In this mechanism, we see in the number of biodegradation level. The extracellular enzyme secreted by the organism inflecting the primary chain to cleave, leading to the formation of low molecular weight fragments like oligomers, monomers, dimers. These are low molecular weight compounds, which can be further utilized by microbes uh, as a carbon asset. Small oligomers might also diffuse into organisms and get assimilated into their environment, so there are some steps of mechanism of biodegradation of plastic. First one is attachment of microorganism on to the surface of polymer. The second one is growth of microorganism using the polymer as a carbon source. Third one is ultimate degradation of polymer. As I have explained before, once the organism attached to surface, it grows the usage of polymer and its carbon supply, and through the process of it secretes some enzyme which break down the dead polymer, and thus ultimate degradation of polymer happens. Next, factors affecting the biodegradation. Biodegradation is governed by mean of different factors that consist of polymer characteristic, type of organism, and the nature of pre-treatment. Chemical and physical homes of plastic play a crucial role in their biodegradation. Side chain owning polymers are tough to degrade, while in the comparison to polymers without side chains, it has been seen that it needs to be saved in the thoughts that polymer with excessive molecular weights are difficult to degrade. The other factors worried within the degradation of polymer are the morphology, melting temperature, and degree of crystallinity. If the polymer is amorphous, then it will be degraded without difficulty as compared to crystalline polymer. Polymer with excessive melting temperature are also hard to biodegrade. Okay, next one we see the five most important eight characteristics which essentially determine the biodegradability of the polymer. The first one is hydrophobicity. The availability of functional group that increase hydrophobicity. Hydrophilic degradation is faster than the hydrophobic, as we know. The second one is molecular weight and density of the polymer. Lower degrades faster than the higher one. The third one is the morphology of polymer plastic. They depend on the amount of the crystalline and amorphous regions. First degrades faster than the crystalline. So the fourth one is structure complexity, such as linearity or the presence of branching in the polymer. Fifth one is the presence of essentially breakable bonds such as ester or amide bonds, chain coupling, uh, like ester to ether to amide and then to urethane. So the next one is the molecular composition. The seventh one is the nature and physical form of the polymer. Films like films, pellets, powder, or fibers. So the last one is hardness. Soft polymer degrade faster than the harder one. So the next slide is microorganisms involved in degradation. There is a lot, many organisms which are involved and have mechanism to degrade and large and complicated hydrocarbons into simple biomolecules. Here we mostly talk about bacteria and fungi, some species of bacteria and fungi, and in species we include gram-negative, streptococcus, staphylococcus, and micrococcus. And in gram-positive, this include Moxella, Pseudomonas, etc. And in the fungal based species of fungus region is include basically asparagus, we then further include asparagus glapus and asparagus niger. In addition, bacillus eutropha, azubecter, and halomonas species are also involved in this breakdown method. Plastic degradation pathways in bacteria. Synthetic polymers serve as a nutrient, as a carbon and energy source for heterotrophs such as fungi and bacteria in many ways. And it basically includes polyethylene, tetraphthalate, polyurethane, polystyrene, low density polystyrene, ethylene, high density polyethylene, commonly found in the agriculture soils as microplastics. When microbes don't get into contact with plastic, oxygen, and ultraviolet radiation, these are the most crucial determinants that initiate the chain reaction in the carbon-carbon backbone. Shorter polymer fragments or oligomers generated during the process are susceptible to get attacked by microbes. Therefore, biodegradation is proceeded over the biodegradation. Uh, Synthetic polymer are such as homoheteropolymer, which may contain same kind or different kind of monomer. The degradation process is achieved by the microbe having different bond, cleavage, and enzymatic activity. Thus, the microbial attack on the polymer surface can be direct or indirect. In the direct mechanism, microbes attack and degrade the polymer for its nutrition and growth. On the contrary, in the indirect mechanism, metabolic products produced by the microbes degrade or deteriorate the polymer. It occurs in a consecutive manner where physical and chemical traits of the polymer are alternated, like biodeterioration, followed by the enzymatic cleavage, fragmentation, then assimilation and the mineralization. Here, so we they see the two pathways in metabolic activity. The first one is the natural metabolic pathway. Depolymerase enzyme are mainly employed in plastic degradation. Extracellular enzymes are secreted by microorganisms which cleaves complex polymers to their corresponding monomers and dimers. These generally undergo hydrolytic cleavage in the periplastic space and the cell membrane. If we go in detail in the natural metabolic pathway, we see that when enzymatic activity involved, they are further exploited as carbon and energy sources by the intracellular enzymes. Oligomers can be directly internalized and presumably with the aid of biosurfactants produced by the microbes. Thus, entering beta radiations can be further cleared by the abiotic processes before internalization. Biosurfactants are produced during biofilm formation. Alternately, these monomers can also undergo subsequential degradation into the common metabolite of the TCA cycle and enter into the central carbon metabolism. It has been reported that the appearance of acetaldehyde, pyruvate to vinyl muconate to phenyl ethanol during the biodegradation of styrene. These compounds are further metabolized to phenylacetyl coenzyme A and thus enter into the central carbon metabolism at the tricarboxylic acid, TCA, which is also known as TCA cycle. Next one is the engineered pathway. During the degradation of homopolymeric plastic material, one kind of monomer is being produced, which either undergo the beta-oxidation or the TCA cycle. Like generally, the complete degradation pathway genes are complemented by engineering different bacterial species. Additionally, a European website has reporting during the PET degradation. Associates has synthesized an enzyme which hydrolyzes the polymer to yield the tetraflate and ethylene glycol are two principal monomers. That is the first step of the biodegradation of the engineered pathway. Polymer harboring hydrocarbon chains are degraded by different enzymes, commonly termed antipolymerase, obtained right from the commonase enzyme, and the uh, other different enzyme is a toxic. They are toxic molecules that are related to this enzyme. Consequently, this is utilized by a root which creating various dioxinases to utilize this to as a nutrient source the, such that it detoxifies that enzyme and the byproducts and thus accept this the less harmful products is produced along with the carbon dioxide and the water byproduct. The next one is biofilms forming microbes in degradation. As we have talked about bacteria and fungi before, that they are basically involved in the degradation of plastic. Microbial community accumulate on the solid surface, therefore by forming the biofilms. Here first we see about the bacteria. So around 90 microbial genera was reported to degrade plastics. Microbial community composition varies in region. It has been suggested the involvement of Pseudomonas and Rhodocus species along with fungus strain which can be used, except that there are many species which are used in this process. Here we see some species and their action on the different polymers like Clostridium botulinum, which act on PCLs, firmicutes, which act on PHP, PCL, and PBS, like polybutylene succinate, and Pseudomonas, which act on polystyrene acetals, polyethylene tetraflate, polyvinyl chloride, polyethylene include, and also include firmicutes again, and it includes different five, and then Pseudomonas and uh, which include it's three different more genera which again act on polyhydroxyalkanone, etc. These are some species of bacteria which has been involved. And the next we see the fungi. Fungi basically include the asparagus, nuclear, and penicillium species. And except this, it also include uh, uh, Rhizopus species and Fusarium, which act on PCL and PHP and PBS. These are some fungus species which act on the polymer in different way, like the bacteria has been acted on the polymer. It is pointed out that the Fusarium species and other fungus species eroded surface and after their attachment. The next one is plastic, biofilm plastic interaction. Plastic was absorbed by inorganic ions and molecules, which promote the microbial attachments. If attached microbes initiate hydrophobic interaction upon its contact with the polymer surface, it is observed that the elevated bacterial numbers on PVC and polyethylene than stainless steel, which they speculated to the leaching of additives served as a possible nutrient source. Polysaccharides and the nucleic acids of EPS secreted by the initiator organism adhere to the film are known to be relatively sticky, which are conditioned to film. It facilitates the colonization for the other organism. Furthermore, file conditioning customized community colonization by governing material specific surface attributes resulting in the leaching of carbon compounds. So the last one topic is bioremediation. One of the most recent ways to dispose plastic waste is bioremediation, which is the removal of polluting substances from an area through the microorganism. While recycling and using bioplastics can be efficient method to prevent waste from being landfills, Bioremediation can solve the problems of environment that are already polluted. Microbial degradation can be studied in various ways, of which one can lead to the different results and has its own pros and cons, and the productivity of experiments and the reliability of the results. Here I have written two terms: polyurethane and endothic fungi and pet and bacteria. If we talk about the first one term, polyurethane and the endophytic fungi. Here, some report in 2008, a group of researchers from the Yale University tested the ability of various organisms to degrade polyurethane. 59 endophytic fungi were grown in the following PUR medium, like polyurethane medium. In the first assessed, 18 of these organisms produced as a hollow of clearance on the surface of the growth medium. Therefore, they had been screened in other assays. Okay, in the first one, all 18 fungi could further examined in other assays. These other experiments showed that the microspora and the other genera of microspora are the quickest to degrade PUR as the sole carbon source in the liquid cultures, and the two that are able to grow in the anaerobic conditions. As with fat and bacteria, if we talk about this as with polyurethane, various studies have been carried out about the microorganism which can degrade polyethylene tetrafluoride. In 2018, one of the most significant experiment was performed by Dr. Yoshida and his team, who screened 250 plastic samples to identify pet degrading microorganisms. In this way, they discovered a new species which was able to degrade a film of 0.31 milligram per cubic meter in per day. So the biofilm community, next one is conclusion. In conclusion, we see the microbial degradation is better than the physical and the chemical methods at the degradation pathway led to the complete degradation and mineralization of polymer. Utilization of molecular techniques to detect specific groups of microorganisms involved in the degradation process will allow better understanding. So that biofilm community play a significant role in modifying the physiochemical properties and degradation of plastic. As biofilm offers bioavailability of nutrients, ensuring a metabolism without the accumulation of metabolic products, resulting in the increased cell viability and degradation efficiency. So at the end, I want to say that, however, biodegradability depends upon the microbial biofilm community adhered to it. The characterization of efficient plastic degrading microbes at the molecular level is still not available. So the researchers could be focused in the field of genomics and proteomics, which could speed up the degradation. That's all about my topic. Thank you very much.